Okay, next speaker is Professor Jack Gallant from Berkeley. There may be a variation in the title.
Since I think you made my title up for me, yeah. There might be a variation in the title. Okay, can we make that, like, big? I feel very insecure because usually I do everything with my own laptop, and I'm using PDFs today, and I'm just not used to it. I use Linux, and that way they can't usually make me use somebody else's laptop. All right, I'm going to talk about reproducibility, and specifically not really reproducibility, I guess replicability, as Martin said. And I think we do have a serious problem, but there's a fairly straightforward solution which has been adopted in other fields, and I think it's time for neuroscience to move to that solution. And what I'm going to say, much of what I'm going to say is going to repeat what Martin said, but using different words because I'm not a real statistician, I'm just a fake statistician. No, not that one. Which button is it? Oh, it's the big button. Okay, the big button. All right. So obviously we have some sort of problem with reproducibility. Everybody's complaining about it, and in fact the public has noticed, and the public's already pretty angry at fMRI these days. So when the newspaper starts saying not only does MRI suck, but it's not even reliably sucking, then you have a serious problem. We've got to do something about that. Otherwise, eventually the government's going to stop giving us money. Effect size, the real problem here is that people tend to study small effects because people, scientists have an itch they're trying to scratch. And that itch may be a big itch to you, but it may be a small itch overall. And so lots of times the things we tend to study are the small effects because somebody else already got the big effects. The low-hanging fruit has already been picked. And so we end up working with these small effects. And when you have small effects, you're going to end up working at the margins of statistical significance, and they're going to be hugely affected by the two variables that affect significance that we all know about, which is the number of subjects we have and the signal-to-noise ratio. And in MRI in particular, we always have problems with signal-to-noise because MRI is an absolutely horrible way to measure the brain. It's just better than everything else. But you figure there's 80 billion neurons, and you've got there's 20 billion neurons in cortex, and you've got 100,000 voxels. There's obviously a lot of data lossage that's gone on, right? So basically, we've got a situation where we've got mostly type 2 error, and in that situation, there's going to be serious problems with replicability. So for all these reasons, it's really critically important to optimize everything in the experimental design, the preprocessing pipeline, the experimental design, the data analysis, and everything to eke out more signal-to-noise. So I think there's 4 or 5 different problems that cause problems with replicability. One is the old method delivered to us by Sir Ronald Fisher and used by psychologists for the past 100 years, which is the use of a single data set for both model estimation and model validation. The second is an over-reliance on significance. Remember, significance was never supposed to be the end result. It was just what you did to make sure your data wasn't random stuff. Suboptimal data acquisition, suboptimal data analysis, and then these unmeasured and uncontrolled state variables, which cause horrible problems and dominate certain things like resting state data. So I'll go through each one of these as best I can. So I'm not picking on Nancy Canwisher here. I'm showing this slide actually because this is a study I think that launched 1,000 ships. It's Nancy's famous face-place study that showed that you can kind of adapt the normal psychology way to do additive and subtractive factors logic into MRI, and you can have a couple conditions, and you can collect time series, and you can subtract them with t-tests and do a little smoothing in SPM, and everything will work, and you'll end up finding areas that you might want to make conclusions about. And this is a perfectly fine study. There's nothing wrong with this study, although even Nancy had problems with signal-to-noise. If you recall, initially when these studies were reported, it was thought that FFA was only, it was not bilateral, and in fact FFA is bilateral, but it turns out that the vein size varies with handedness. MRI is affected by vein size, and so you end up having a bigger FFA on one side of the brain than the other side of the brain. And so the signal-to-noise is different in one side of the brain than the other side of the brain. And so if you don't have good enough signal-to-noise, this phenomenon will look unilateral when in fact it's bilateral. Now people have come up with a lot of methods to try to get around the problem with statistical significance testing, which is the really serious problem that we have, right, with multiple comparisons. And FDR correction is the standard way that people have started to use. Everybody, basically, I think in every paper I read, uses FDR correction. We use FDR correction when we calculate statistical significance, and there's nothing wrong with it. It works great. However, it doesn't solve the problem of type 2 error. It doesn't do anything about signal to noise. It's just a way of making fewer mistakes when you're calculating statistical significance. And remember, the point is not to calculate statistical significance. The point is to build a model that explains the data and accurately predicts new data sets. So it's interesting that I deal a lot with computer vision people because I'm kind of at the interface between human vision and computer vision. And computer vision, about 10 years ago, had a really serious problem. And it was caused by it was caused by the fact that computer vision people don't like to collect data. So they would come up with an image set. Say I'm going to make an image set of 10 cars, and then I'm going to run my computer vision algorithm on these 10 cars. I'm going to show that I can perfectly accurately predict what kind of car is in each of these 10 images. And all the computer vision people did this, and every computer vision algorithm that was ever reported looked like it worked fantastically. And as you know, we still have no robots that can see. That's because computer vision is really hard. And people were essentially overfitting to their tiny data sets. Every single individual computer vision researcher was collecting his own set of images and then just overfitting the heck out of them and making everything look perfect, and nothing generalized outside of set. So the computer vision community got together and they came up with two solutions to this problem, two integrated solutions, and I'm only going to advocate that we adopt one. The first solution is they came up with giant data sets at a community level. And the big one that people know about right now is called ImageNet. ImageNet has thousands of images in it. It was an image database collected by Fei-Fei Li at Stanford. And these image databases are used by everyone to fit their models. And the databases are large and diverse, so nobody has to worry about overfitting to a small data set. The second thing they did is they gamified things. So they said, okay, we're going to give you a bunch of images, and you can fit whatever the heck computer vision model you want on it, but we're going to reserve some small set of images that we're not going to give you. And now you give us your computer vision algorithm, and we're going to run it on this small set of reserved images, and we're going to calculate how well your computer vision algorithm does on this separate set of images. And this prevents people from overfitting and cheating, and they're not responsible as individuals to keep from overfitting and cheating. They don't have the possibility of overfitting because they don't have this separate test data set. So the first data set that you fit your data on in computer vision is called a fit set, and the second set is called a test set, and this helps keep everybody honest and has led to the remarkable improvement in computer vision over the past 10 years. So I'm advocating that we adopt the fit and test set paradigm in our field as well. In my lab, historically, we've always called this an estimation validation paradigm rather than test and fit, and because it conflicts with everybody else's terminology, I'm kind of sorry we ever started using that, but that's what we did. The estimation data are used to fit your model, and the validation data are used to test statistical significance. Because the fMRI responses are non-stationary, and everybody pre-processes their MRI data and spheres the heck out of it and makes it Gaussian, it's very important to collect these two fit and test data sets at the same time in some sort of interleaved design so that you won't end up with problems of essentially different variants in the two different data sets. You also have problems with adaptation that I'll get to in just a second. And there's a whole bunch of issues that come up with optimal stimulus design in this kind of paradigm, but the real thing you just have to keep in mind is collect two data sets. And even if you don't want to collect two data sets, you can just collect one data set and then divide it into two parts. That'll work the same way. So in my lab, we tend to use natural stimuli rather than a controlled experiment. So we collect a very complicated data set. For example, people watch natural movies, and then we'll fit 20 models to those data, and each model is essentially a hypothesis. And this is a slightly different procedure than the usual, which is where you optimize your data for one hypothesis. Does this thing actually have a laser pointer on it? The red button. Oh, a very dim red laser pointer. Okay, well, that's better than nothing. Okay, so we take our data, our estimation stimuli and our validation stimuli, and we interleave them. So now we've created this long sequence of stimuli, and we collect our MRI data, and that's going to give us a long sequence of responses, which I've just colored here red and blue so you can keep track of what's going on. We're going to pre-process our data, which in MRI usually means making it Gaussian, throwing away anything that's not Gaussian. It doesn't matter if it's signal or not, you throw it away. You take the Gaussian stuff, that's what you're going to analyze. Now you break these data up into two sets. A fit set, which you're going to use to fit your models. And in our paradigm, we, in the fit set, collect only one observation of each stimulus because we want to sample the stimulus space as largely as possible, as broadly as possible. And then we take these different data up here, the test set, and in those data, we've repeated them. So because we've repeated them, we can calculate the mean, and we can calculate the standard deviation. We can calculate the standard error, and then we can compute a noise ceiling. So in the validation data, we're actually going to be able to say, in the absence of any model, how much of the variance could the optimal model ever predict? And that gives us a noise ceiling. That gives us a target to shoot for. So now we estimate a GLM on the fit data, and we're going to get a bunch of beta weights out of that. And then we're going to take those beta weights and we're going to apply them to the test data. And we're going to get predictions, and we're going to compare the predictions to the actual observed data, and we'll do as well as we do. If our noise ceiling says that the optimal model should predict 70% of the data, and our model predicts 50% of the data, we know exactly how good we're doing. We're doing 5/7 of science. When we get to 7/7, we're done, right? So you know when you're finished here. Yeah, you compute statistical significance along the way, because if you don't, the reviewers will yell at you. But really, nobody cares about significance. It's all about predictions. The whole point is to predict as much of the variance as possible. So you can think of the variance this way. There's a total possible variance in the phenomena as a whole that that the best model could ever account for. Now, you're not going to rove the entire regime of possible phenomenon. For example, in movies, you can't possibly test all possible movies. So in your subset of movies, there's some amount of variance you could potentially explain. But your finite data set is limited, so that actually means there's going to be less variance you can explain. And then this red ball here is the actual variance that you did explain, and this little tiny black ball is statistical significance. So you can see things get worse and worse and worse. What you want to do is basically explain all of this blue stuff here. So if you're doing a standard experiment, you can do the same thing, except here the red epochs are repeats, and so you're going to stack them all up at these two stages. But still, you're going to break your data up into two segments. Now, this is a standard significance effect. Again, I'm picking on Nancy here, but it's actually because this is one of the best studies ever done in language. It's the first single subject study of language ever done, and you can see that you get significant results for language for this subject. But if you use a voxel-wise modeling approach where you look at predictions rather than language and do a few other post-processing things I'm not going to talk about here, you actually get way better data. You get, in this case, you're getting less than 1 bit per voxel because you're essentially just doing a statistical threshold, which is a yes-no decision, and you've smoothed because that's what SPM wants you to do, so you're getting less than 1 bit per voxel. In this paradigm, you can get up to 6 to 10 bits per voxel. So you get a lot more mutual information between the experimental stimulus and task conditions and your dependent variable. Now, there's a whole bunch of things you can do to screw up your data, and I would just suggest that you try to avoid them as much as possible. Grappa is evil because it violates the first law of science, which is that there's no free lunch. It appears to give you a free lunch, but it doesn't. It requires scout images, and your subjects always move, and so it always screws up your signal to noise. Multiband is somewhat evil, only partially evil. It's actually good if you do a little bit of multiband. If you do multiband 2 or 3, the signal quality is exactly the same, but the noise goes down, so your signal to noise ratio goes up. If you do multiband beyond 3, then your noise starts to go up again, so your signal to noise ratio starts to go down. So a small amount of multiband is actually quite good. A large amount of multiband is bad. There are a whole bunch of other decisions that will change the signal to noise in your experiment. The pre-processing pipeline will change your signal to noise in your experiment. If you don't use field maps, it's going to be very, very difficult for you to correctly shim and correct for the bad shimming of your magnet and the fact that you have a solid body in the middle of your magnetic field. The unwarping algorithms matter a lot. If your subjects moved or you didn't do motion correction correctly, bad things will happen. Your co-registration is an area fraught with danger. Reslicing decisions oftentimes screw up your data and reduce signal to noise, especially if you reslice more than once. You almost always have to reslice once, but you want to reslice as little as possible. And of course, the detrending algorithm matters a lot. You can linearly detrend, which is horrible. You can do polynomial detrending. The best detrending that's easy is usually median detrending. You can use a polynomial median detrend called a Savitsky-Golay filter that works really well. Or you can do what the people at NIH do now and use white matter to come up with a detrending kernel, which actually works best. So the biggest effect on signal to noise that you have in your, can you tell me how long I have left, Victor? Oh, I have a clock. Wow. If I wore my glasses, these things would be obvious. Okay. So, so the biggest effect that you have on signal to noise in your magnet is head size. That's the number one thing. What you ideally want is a subject whose brain is right next to the antenna. But the coils that Siemens gives you, Siemens is a big company, they've got to sell a lot of these things. They don't want to make coils for everybody because they're very expensive. So they make coils that are large enough to fit 95% of human heads. That's a large number of human heads, many of which are very big. So that means that if you take a generic human head and put it in the middle of that coil, the brain is going to be very far away from the coil. And that's especially going to be true if you have a subject that has a thick skull. Ideally, the best possible MRI subject would be somebody whose head is exactly the same shape as the inside of the coil, and they have an infinitely thin skull like an eggshell. That would be a perfect subject. Few of those people exist. So, you're...
A large infant.
A large infant, yes, a very large infant, exactly. So generally your data is going to be bad, and this I'm just showing you here the predictions. These are predictions, essentially just percent variance accounted for in the cross-validated data set in an attention experiment. And you can see 4 different subjects here, and this is just the standard heat map. So yellow here is very good predictions, and gray is horrible predictions. And you can see that the prediction quality here varies hugely across subjects. And this is due to 2 main factors. One is head size. You always want subjects with big heads. And the second is who ran the experiment. Because all of those who who've done MRI experiments know the best data always comes from the person who's got the biggest vested interest in the experiment working, because they're paying attention and they're motivated. And the other subjects really just want to get paid. So, so this is a serious problem. Obviously, incorrect alignment, if you decide to aggregate these data together, is also going to reduce signal to noise. Remember, alignment is not a solved problem. You can do volumetric alignment with affine transforms, or you can do surface-based alignment like FreeSurfer does, but none of those methods are optimal because they're all based on anatomy, and the correlation between anatomy and function is not perfect. And in fact, the correlation between anatomy in the small sulci rather than the major sulci is very difficult to know because people fall into different sort of subsets of classes in the arrangement of their anatomical sulci. So FreeSurfer or the volumetric alignment programs have serious problems aligning the volumes, and the alignment just isn't that good in the end. So that also decreases signal to noise. Signal to noise is substantially affected by attention and motivation. Oh, I said this in the last slide. Did I talk about the wrong thing in that slide? Right. A few other things on here. Brain symmetry, right? If your brain is symmetric, you're going to have better signal than if your brain is asymmetric. Right. Okay, so these are attention maps. This is from an experiment where people were doing a simple attention task. They were tending to cars in movies, or they were tending to humans in movies. Vehicles in movies, or humans in movies. And we collected data under these 2 conditions, and we fit a 985-dimensional semantic model to these subjects' data under these 2 conditions. And then we found a common semantic space for all of the subjects, and then we projected the voxels into this semantic space. And essentially, all you really need to know about this is that the individual colors on these cortical flat maps indicate the semantic tuning of individual voxels. And in this particular map, as I recall, red means more motion selective, and yellow and green are animal and human selective, and bluish is more selective to indoor and outdoor scenes, or more tuned, I should say, rather than selective. And you can see that there are similarities between subject 1 and subject 2. The top here is the baseline map, where the subject is not attending to anything. The middle here is the map where they're attending to humans and animals, and you can see that the human and animal representation is increased. And the bottom is the map when they're attending to vehicles, and you can see that the representation of outdoor scenes is increased. Now, I should mention that these attention results were obtained when the people were looking for people, humans, or vehicles, but when there were no humans or vehicles in the movies. So this is a preparatory response. These are attention changes due to anticipation and target search rather than target detection events. But the point here is that the left hand and the right hand columns are somewhat different. These 2 subjects have very different kinds of representations. There are largely similarities, but also substantial differences. And if we just averaged all these data together before looking at it at the individual subject level, we would lose a lot of information. We'd lose signal to noise. The individual differences would average each other out, and the only thing we would find is the variance in common. And this is one reason why when we see published MRI results, generally they look very punctate. That's due to statistical thresholding and due to aggregation of the data at the cross-subject level before the individual data from the individual subjects is analyzed. There's a general rule in statistics, which is you should always use the simplest model first. And the simplest model, for example, is a univariate model in the brain. If you want to look at covariances, say using resting state, that's a second-order model. If you want to look at individual subjects, that's an easier, simpler model than a cross-subject model because a cross-subject model involves more assumptions. A very common thing that we see in MRI, which doesn't directly affect repeatability and replicability, but which contaminates our conception of what our findings mean and therefore affects repeatability, is incorrect logic concerning effects. The thing to remember is MRI data is mostly type 2 error. There's signal in there that we can't see. The signal to noise is very low. Therefore, it's critically important to remember that if we don't see an effect, we can't say anything. We are not allowed to ever say anything about an effect that's absent. And this is particularly problematic when people compare across areas. So the data here at the bottom are data from a language study where people basically just listen to stories, and then we fit a semantic model to the language data that we got. And this is, again, predictions of where the language model fits well on both the flattened cortex and on three-dimensional volumes. Now, if you're a neurolinguist, you remember that there are kind of three parts of the brain where if people end up with dementia, it strongly affects their language ability and their semantic processing. We've got the Broca's area up here, and we've got Wernicke's area here. And then there's this anterior temporal lobe where it's known that if you get dementia in the anterior temporal lobe, it screws up your language. Now, you may notice we don't have any semantic model fits in the anterior temporal lobe here. So that seems on the surface of it to conflict with 100 years of neuropsychology, and that would make the neuropsychologists unhappy, and that would probably mean our reviewer would yell at us. So why do we have this problem? We have this problem because there's just no signal there. Nobody in MRI records signals in the anterior temporal lobe unless they specifically optimize everything to record signals in the anterior temporal lobe because it's down by your ear holes, and MRI signals are poor. So you can't make any comparison with signal in the anterior temporal lobe to signal in Wernicke's and Broca's areas because you just have no signal there. The signal to noise is different. And this is a general rule. You can't actually ever compare across areas unless you have normalized for differences in signal to noise. It's logically incorrect to say, I found a result in area A, and I didn't find a result in area B, and therefore, shmoo, unless you have shown that area A and area B have common signal to noise, or you've built a model that somehow normalizes for the differences in the signal to noise. And I personally have never seen an MRI study that did that, and I think everybody should do it. We could talk for a few seconds about resting state, since I have 1 minute and 59 seconds left. Resting state is an interesting phenomena. It's obviously real data. However, I'm personally not a big fan of resting state because I think, again, it tends to look like a free lunch. It's easy, resting state. You put somebody in the magnet, they just rest. You get all the data you want. What could be the problem? But resting state, really, in some sense, it's a blind estimation problem. Really what you want to know is you want to know how the brain represents either the world or internal states. So you want to find the relationship between the variables you measure, which are MRI signals, and these states of the world or these internal states, these X variables. You want to find the relationship between X and Y, and you want to build a model, and that's going to be science. That's going to be your explanation. But the problem in resting state data is you don't have the X's, you only have the Y's. So now you have this modeling problem where you have to model the relationship between the X and the Y's using only the covariances between the Y's. That is a really, really hard problem. And I think it's so challenging that it's not one I personally want to deal with. Now, more recently, people have used not resting state, but covariance-based analyses, because all the resting state data in MRI are all covariance-based analyses, since the data's all become Gaussian. People have used task-based network analysis. And in that situation, you're better off because you have an X variable, which is your task, and you have a Y variable, which is the covariances between your voxels. And so it's a perfectly reasonable statistical problem to try to find out the relationship between the covariances of Y and your X variables. However, remember what I said earlier is you always want to fit the simplest model first. So if you're going to fit a task, if you're going to analyze task data, with a covariance-based analysis, my opinion is the first thing you need to do is go into the task data and fit a simple task model, a first-order model, subtract that off, and then take the residual covariances and model that. Because that way you're sure that you're not modeling in your covariances the first-order signals. If you don't do that, then those first-order signals are going to be variants that will be modeled in the second order, even though that's not correct and you'll be making an incorrect conclusion.
No overfitting, Jack, but over time.
I'm done. This is it.
All right.
I've got 18 seconds left.
No, that's going negative.
Oh, it is. You're right. Okay. Well, then I will quit. Thank you.
Okay. All right, as usual, Jack Galland has given us a ton of stuff to think about. We run out of time, so we'll move straight on to the next one.